The reason why I'm showing you that DNA modifications are predisposing to melanoma, this is important because I'm going to tell you how we try to make melanoma in mice. Okay, so now I told you that melanoma is in the skin, but where is melanoma coming from? The melanoma is coming from the transformation of a cell that is called melanocyte, and melanocytes are skin cells which give you pigment. And in fact, the melanocytes are located here. This is a section of the skin, human skin, and here this is the epidermis, and this is the dermis, and here is a layer called the dermal epidermal junction, and this is a hair, and this is the hair follicle. So what you have is those are melanocytes here, and generally you have the melanocytes are sitting in the dermal epidermal junction, and they will emit dendrites here. Shown. This is a melanocyte. Those are the dendrites, and they will connect with the keratinocytes, which are the cells of your skin. And what they are going to do is they are going to produce the melanin, which is the pigment color, and then you will tan. So when you go to the sun, what happens is that your melanocytes get activated, they produce melanin, they give them to the keratinocyte, if you will, and then you have a darker skin. So having a darker skin doesn't mean you have more melanocyte, it means you produce more melanin, okay? So what happens when you have cancer? When you have cancer, you have transformation of the melanocyte. So basically the cells we just saw before start proliferating and they grow like crazy. You see, here they are proliferating, and here is an example which we call the radial growth phase of the melanoma. And then there is what we call the vertical growth phase in melanoma, which is worse. So the cells start invading even the dermis, okay? And then after that they start metastasizing and it goes everywhere in the body, okay? So this is just to recapitulate one thing, how a melanocyte becomes a melanoma, and also to tell you that this is a multi-step process. Okay, so first of all you have this name here is the name of a mutation. This is the name of a gene. So when this gene is mutated, for example, the melanocytes which are in the skin start proliferating. Here you have shown doubling, okay, and here they proliferate even more because you have other mutations that are showing up in this person, and then you have even more mutation, and you have the vertical growth phase and changes in other genes, and then you start having independence of growth factor, and the cells really they metastasize and they go to other organs. So what you have to remember is the melanoma is coming from melanocytes, which are normal cells, when they are transformed, and this transformation is a multi-step process. It doesn't happen all at once, it happens in multi-steps, okay? Okay, so here, this is a little bit scary, but this is the picture of a pathway, and this is what happens in the cell. Okay, so you have, here is shown as an example, for example, there are two very important genes which are mutated, which happen to be mutated in some melanoma, and this is the P14 gene and P16 gene. And these genes, or the product of this gene, because this shows a protein, will interact with other proteins which are P16, cyclin kinase, cyclin D, RB, and I don't know, and you have overproliferation of this melanocyte or any cell type. Okay, and this is P53 damage. So here, if you have mutation here, then you perturb the P53 pathway. So this is, so the take-home message is that if you perturb a pathway which is important for proliferation or protection for DNA damage, you might get cancer. Okay, so now, in order to do this, we want to model the cancer in mice. But why do we use mice? We use mice because they are physiologically similar to humans. Yes, they look very different, but believe it or not, they are still very close to us. And also because of all the recent advances in technologies which have developed that area, also the development in the genome study of the mouse genes. This helps a lot in making this tool something easier to use than any other animal system. Okay, So what goal can be achieved by manipulating mice? Well, we can do genetic engineering of a desirable trait, means we can put in a mutation for a certain disease. We can create a rapid creation of pure breeding line and identical offsprings, which are the clones. We can do animal model system to research in human disease, which is kind of repetitive for this. And they can be used as animal bioreactor. They can be used to produce some stuff which we want to use, and what this is called animal farming. Okay, so how do we do that? How do you make animal models of transgenic or knockout animals?